I thank you for being here. My name is Ana Morales, and I'm a CEM fellow. Um, Over the next 40 minutes, I would like to discuss with you the population genetics of human pathogens, particularly focusing on tuberculosis, influenza A, and cytomegalovirus. I would also like you to wait until the end of the presentation for the questions. I have a lot of windows here, and I'm afraid I might be confused if I have many people asking me things at the same time. So yeah, please write down your questions. I'll be really happy to talk about them at the end. And I will remain a little longer with the students if you are there. At the end of the presentation and the talk, I will join you for another extra brief discussion. First, I would like to give some, not scrolling down. I'm sorry, some technical issue here. Okay, I would like to give you some generalities about these pathogens and the infectious diseases that they cause. First, tuberculosis, which is caused by a bacillus named Bacterium tuberculosis. It usually infects the lungs, and it causes about 10 million new cases per year worldwide, but it's mostly in Asia, Africa, and the Middle East, the highest burden now. Even in the US, there are about 10,000 cases per year, and it kills about 1.5 million people per year, meaning like there's about 4,000 people dying every day of TB. Many of them are children, which is 250,000 of children dying of TB. It represents an important challenge because of drug resistance, which globally it's about 5%. And last year only, there were about 500,000 new cases of people resistant to antibiotics for this disease. Another case is the influenza A virus, which is a single-strand RNA virus. I will be talking here only about the H1N1. It also has a high impact in human health with about 600,000 cases of death per year. And this is only talking about the respiratory failure and not with any other complications that could be derived from the infection. It has a low rate of drug resistance, only 1 to 2%, and different from tuberculosis. This influenza could have and can have animal reservoirs. Mycobacterium tuberculosis, it's only infects humans, and we are the reservoir and the ones that also get sick. And finally, the human cytomegalovirus, which is also a human-specific pathogen. It's a double-strand DNA virus, and it's the largest of the herpesviruses. It is very prevalent worldwide. We all have it. It is transmitted by contact with any secretion or body fluid from tears, blood, urine, breast milk, semen, everything. And it's very challenging also sometimes for organs transplants. It's considered an opportunistic pathogen because it affects mainly people who are immune compromised and also neonates that have less developed immune system. It's the highest infection that occurs in neonates, and it's about 3,000 of them per year only in the US. It has not a specific treatment, and the consequences of these infections are really serious, going from blindness to deafness to brain damage, etc. Though these pathogens are very different species, they share, or we share, the same questions for them, which is from the population genetics perspective, what are the underlying evolutionary processes dictating the within-host genetic diversity? With that, I refer what are the relative roles of genetic drift or transmission bottlenecks, versus purifying selection and background selection versus a spatial subdivision or compartmentalization. Because then they can all develop infection in different parts of the body, and that's also important for genetic diversity, maybe. And another question that we have from the population genetics perspective is if these pathogens represent examples of the Ryan-Fisher model violations. Ryan-Fisher model is a widely used null model in population genetics since 1930s, under which, among other assumptions, their offspring is small in all individuals. However, we know that there are some organisms that, virtue of their reproductive biology, produce a large offspring that has been observed in sardines, in oysters, and is also very clear in viruses such as influenza virus. And with that, in these cases, there is probably a better coalescent model, which is the multi-merger coalescent versus the Kickman model that derives from the Ryan-Fisher model. And in this case, it allows that a large proportion or a significant proportion of organisms descending from one single individual every generation can be so important that could change significantly allele frequencies. We are going to apply, or we have applied these questions to Mycobacterium tuberculosis, and this is the first case example I'm going to give you. How does it work? How do we address these questions theoretically, and what are our results? First, we consider the main parameters for genomes in these populations. For example, we know the Mycobacterium has a small genome size. This specific Mycobacterium tuberculosis has a small genome size of only 4.5 million base pairs. It is highly coding, so more than 90% of the genome is coding, and it's extremely clonal. So there's no recombination, no horizontal gene transfer. So it develops antibiotics resistance very quickly. The generation time or the reproduction time is between 15 to 24 hours. And it is unclear the mutation rate. In vitro, it has been measured to be in the order of -10 per site per generation. But when we have experiments using non-human primates models and use antibiotics and trying to measure these mutation rates, it clearly rises up to 10 to the -9, 10 to the -8, and 10 to the -7. So it's a broad range of mutation rates that have been described for this pathogen. And another important feature is that Mycobacterium tuberculosis, usually their lineages correlate with certain geographic areas. Some lineages are more common in the Americas, in Europe, and others in Asia. However, every of these lineages, or each of these lineages showed here, represents a large aggregate of individuals summarized in one consensus sequence from a clinical sample, meaning there is a bunch of variation that we don't detect using consensus sequences and to come within the host. So this is why we want to switch a little into a perspective from within the host and try to observe the variation from that. Previously, and thanks to the next generation sequencing, we know that most of these variation that we observe within the host are compounded by rare alleles. And there is a low number of segregating sites. For example, in this publication from 2017, in which they sample patients since the moment of diagnosis and through the course of treatment every 2 weeks, they observe 0 segregating sites or polymorphism at one time of sample, and they could observe also up to 50 segregating sites from another patient. And important to say, regarding the number of circulating sites, it does not correlate with the success of treatment. So moving forward and thinking about what are the evolutionary processes reducing the genetic diversity that we observe in these populations, we consider transmission bottlenecks. At the moment of transmission, the infection can be established by only one single bacterial. And that for sure will reduce the genetic diversity from one population that lived in one patient into the new one. And we also have clonality that we also know that reduces variation and the effective population size. Another aspect that has been very well studied in mycobacterium tuberculosis is the effect of purifying and background selection, because it is clonal, so the effects of purifying selection is expected to be very high. So purifying selection is when a deleterious mutation, here represented by the red dot, present in one genome, it's eliminated and it's not present in the next generation. But it also eliminates all the sites that are non-deleterious linked to this one that is deleterious, and that is the background selection. And then finally, we have progeny skew, in which we know it happens in other pathogens in which there is clonality, and some of these bacteria can have a very large number of descendants, and therefore multi-merger coalescence could be a good option to explore with it. In order to summarize all the questions that we had about the within host mycobacterium tuberculosis evolution, is basically if these populations are evolving under non-Rhino-Fisher model, if the neutral processes that we know can explain the population's diversity, if we can detect the infection bottlenecks, and if these infection bottlenecks, how hard are these affecting diversity, and if we can actually infer the progeny skew in these populations, and also the mutation rate in the presence of all the rest of the processes. In order to explore that, what we did was to conduct a simulation study. That the main idea was to develop a null model for this MTB. And in this case, we used two different population sizes, 1,000 and 25,000. We tested populations that had, every individual had 1/10 portion of the real population size. We also considered an equal amount of nearly neutral and slightly deleterious mutations coming into the population every generation. And we tested all the possible ranges of mutation rates, and also different numbers of bottlenecks, reducing the population size to 1, to 2, to 3, up to 100, simulating the transmission bottleneck. And we also included the progeny skew, considering from 0, non-progeny skew happening, up to 2% of the population descending every generation from a single individual. That is considered very high. Even in viruses, that is considered really high. If you think about population sizes of tuberculosis, have been estimated to be in the order of 10 to the 8 or more in a patient. So it's a really large population size, and the progeny skew will be also significant. So after we conducted simulations, we estimated statistics that are considered our priors. And the summary statistics that we chose to work with is the Tajima-T that will reflect the number of red alleles and segregating sites. Then after we had all of these summary statistics for each of these simulations, we compared that with those summary statistics observed in patients. And only from our correlation of results, based on simulations, we saw that mutation rate, as expected, increased the number of segregating sites, but that progeny skew, considering progeny skew, will reduce it strongly, even more than bottlenecks that are happening at one point, while progeny skew could happen every generation. And that therefore will also reduce even more the effective population size. Considering the patient's data, we chose the range of average proportion of segregating sites observed in patients, and that is here highlighted with the orange and blue lines. So that is the range of average proportion of segregating sites that we observe. And the simulations are the dot points, the black dot points, and the replicates are the gray bars. So from here, psi, which is the progeny skew, is in the x-axis. And we separate this by the 4 ranges of mutation rates we were testing. And we observed that most likely mutation rates in 10 to the minus 7 and 10 to the minus 8 are the ones that explain better the observed patient data. Although it looks to the eye that 10 to the minus 9 could have been, we also took note of another observation from our simulation, and is that in our simulation, most of the 10 to the minus 9 mutation rates will give us 0 segregating sites, 0 polymorphisms. And that is extremely too high. That is not observed in the patients. So we decided to exclude that one based on that observation. From there, so we knew that the distribution of polymorphisms in patients, which is here on the top, could be explained, but by the combination of mutation rate and progeny skew. And the extremes are these ones that you see, the left and the right. And if we draw our posterior with this range, we observe that in average, 6 to the minus 8 mutation rate and an average proportion of 5% of progeny skew per generation could explain the data in patients. And that bottleneck is not possible to detect because we think that progeny skew is so frequent and high that it will eventually erase the signal for transmission bottleneck. So we could not infer that. So for conclusions with this approach, we learned that MTB populations are evolving on the non-Writing Fisher model, and that our effective population size is small, and that is due to the reductions of progeny skew, and also which is a product of clonality. And we also found that mutation rates are likely to be much larger than previously believed. And this is important to know because this reconciles the low genetic diversity observation and the rapid spread of antibiotic resistance. Antibiotic resistance can be developed in a patient in a 2 to 4 weeks period. So it's very, very fast. And we also think this is an important finding in order to consider the history of MTB in humans, which needs an accurate mutation rate, and more exactly, the proportion of neutral mutations that are entering in the population every generation, which is needed for estimations of rate of divergence. And talking about that, we move into the next question. So what else do we need to continue to build neutral null models for pathogens? Our main question here is, what is the distribution of thickness effects? What is the S value for the mutations that are coming in every generation? And it's important to know that because we have mutations that are coming in every generation that are advantageous, effectively neutral, slightly deleterious, strongly deleterious, and lethal. And since Ora in 1973, we learned that by the bimodal shape of the distribution of thickness effects, in which most of them are neutral or slightly deleterious. We also know that the distribution of thickness effects is likely to change in time. And the inference of this, which is basically inferred in the background selection, it's important for predicting adaptation. So this is why we are conducting a project on RNA and DNA viruses. This is a project that I am conducting along with my colleague and friend at the Yansi lab, Parujori. And our main objective is the inference of the DFE along with the demographic changes such as phonics and progeny skew. We are applying this to influenza and cytomegaloviruses that we have data available from the Yansi lab. And it is very different one from the other in the sense that for influenza, we are using experimental data. These experiments were conducted years ago, and they were analyzed by Fowler in 2014. In this case, a population of influenza is grown from one single individual up to 10 to the 6 individuals, and then a bottleneck is imposed 12 times, giving them 12 passages or 12 times of bottlenecks. And the idea is that knowing already some of the characteristics of the viruses, such as influenza, that is a small genome, very small genome of only 13,000 base pairs, more or less, packed in 8 fragments that do not recombine, but they reassort very rapidly. And knowing that, so we are choosing one of the segments, which is PB1, in order to apply our method. And in our method, we are considering a mutation rate in the order of -5 and progeny skew of almost 7%. And these are all control lines that have not been exposed to any antibiotics during the experiment. And the progeny skew was estimated by Zuckerman et al. 2019, and it's been observed also directly in the laboratory. We have in our simulations for this model a population size of 10 to the 6, that is the one that the experiment demonstrated, and a small effective population size that was also inferred by Fowler et al. We are using the DFE sampling proportions following Parujori, and so our idea is to find out what is the proportions that we will have after the whole experiment, and what is the more or less shape of this DFE. Then for the cytomegalovirus, we are approaching it from a different perspective, in the sense that we're using patient data here. We are considering some other aspects in these viruses because they are highly variable, so there is recombination, and multiple strains can infect the same patient. They can be highly destructive. So there is a high within host compartmentalization, saying that samples from blood, saliva, and urine from the same patient will differ much more than taking 2 blood samples from different patients and compare them. And because the time of infection is really difficult to establish, because you can get infected at any point of your life, and you don't know when was this viruses starting to infect you, we have chosen newborns, congenitally infected newborns, and using time samples that were already processed from all their compartments. We're using a patient we know that was infected through placenta because the mother was sick, And so we know there is the moment of infection was at the conception, more or less, and that the infection of the kidneys, for example, was at the moment of formation of the kidneys. So there are two important bottlenecks that are here in the red arrow. And in this case, the cytomegalovirus is much larger in terms of the genome size compared with influenza. And we know that it also has a recombination rate that is quite high, 10 to the minus 7. Mutation rate is not as high as influenza. It's only also in the order of 10 to the minus 7. And psi, we're going to use the same that has been observed in influenza and see if that can be the best approach. And using the already estimated effective population sizes from this publication in 2016 and applying also Parul's modeling. So in general conclusions, we think that time sample approach is the best for population genetic inferences because we observe what the changes of allele frequencies by time, and that's a much better approach. It is necessary to develop more neutral null models for human pathogens, the rarely null models for most of the most important pathogens that affects humans. And that multi-merger coalescent, it's a very important framework to try to construct these null models. And it is also an interesting approach, considering there are many other organisms that also don't fit the Reding-Fisher model of spring distributions, including plants or marine organisms and this large variety of human pathogens. And that finally, we should continue to develop a better approach for the distribution of thickness effects, or in bankgraph selection particularly, to clarify the evolutionary processes in pathogens. With that, I want to say thank you, all of you, for waiting. Oh, it's 43 minutes, sorry about that. I'm 3 minutes over. And thank you, the Stone Lab, that I'm part of them, and the Jensen Lab that have been hosting me during my fellowship at the Center for Evolutionary Medicine.